Thank you for this opportunity to present at this webinar. So let me see. Okay. So I've broken this presentation into a few sections. First, I'd like to give you a brief company overview to familiarize the audience with EntroGen so you know who we are and what we offer. And then I will briefly discuss at a high level the evolution of precision oncology in lung cancer, followed by an overview of EntroGen's offerings in this space. And then we will conclude with questions from the audience if there's time. So before I begin the presentation, I feel that I should clarify that when I use the term approval or approved in this presentation, it specifically refers to approvals granted by either the US FDA or the European Medicines Agency. It does not imply that the diagnostic or therapeutic is approved in other regions, such as India. And for the regulatory status in your territory, I encourage you to consult the relevant regulatory authorities or your local clinical guidelines. So as you may already know, EntroGen is an in vitro diagnostic device developer and manufacturer. We've been in this business since 2007, so a little over 17 years now. It is a US-based company located in Woodland Hills, California, which is just outside of Los Angeles. And we offer end-to-end solutions for oncology and hematology. Our product portfolios consists of reagents both for qPCR and NGS assays. We offer hardware in the form of a liquid handler called BenchBot. This is used for nucleic acid isolation and qPCR plate setup. And we also offer software for qPCR and NGS data analysis, which are provided free of charge to our end users to help them with data analysis of our assays. As an IVD manufacturer, we are an ISO 13485 certified company, which is the international standard for medical device makers, and we are also an FDA-inspected GMP facility. So our products are distributed globally. We sell direct in US and Canada, but outside of these two countries, we sell through distributors. Today, we have over 56 authorized resellers that represent our products globally. Most of them are in the European Union, but we also have presence in Central and South Americas, Middle East and Africa, and Asia. We also have a GMP manufacturing facility in Slovenia that is used primarily for our European distribution channel. Our resellers are responsible not only for importation and distribution of our products, but also for any regulatory submission in order to enable their users to utilize the products for diagnostic indications. We also offer training for our resellers to provide their end users with first-line technical support. And we find that this is critical, especially given the time zone differences. So this way users of our products can get support as needed without long delays. EntroGen's focus has always been hematology and oncology. Our real-time PCR and NGS assays are used to identify clinically actionable targets in these indications shown on the slide. And of course, the topic of this webinar is lung cancer, and we have a number of assays on both real-time PCR and NGS platforms that are applicable for this indication. The focus today, however, is going to be on real-time PCR only. And just to give you a glimpse of the products that we carry in our portfolio, as you can see, we have quite a number of products. There are 35 CE-IVD marked products, one FDA-approved companion diagnostic that we received just last year in September of 2023, and a few newly launched research use only kits. But I want to point out here that regardless of the regulatory status of the product, all of these are manufactured under the same quality management system with the same validated manufacturing processes in the same GMP facility by the same trained personnel. So I'll be happy to discuss any of these products if we have time later on, but now let's focus on precision oncology and lung cancer. So precision oncology has really revolutionized how we approach cancer treatment. Instead of relying on generalized chemotherapy or radiation therapy, precision oncology allows us to tailor treatments to the unique genetic makeup of a patient's tumor. This means targeting specific mutations known as actionable mutations that are critical to the cancer's growth and survival. We've moved from one-size-fits-all strategy to a more personalized approach where the right patient gets the right treatment at the right time. The shift relies on advanced diagnostic tools like real-time PCR, which have made it possible to quickly and accurately identify these actionable mutations. In today's presentation, I will explore how this has transformed lung cancer care with a particular focus on improving access to these life-saving technologies. So precision medicine in lung cancer has a remarkable history. In 2004, we saw the discovery of EGFR mutations, which changed the treatment landscape for introducing the first targeted therapies like gefitinib and erlotinib. In 2009, ALK rearrangements were identified, leading to the development of ALK inhibitors such as crizotinib. This was followed by the introduction of KRAS G12C inhibitors around 2015, which addressed a mutation once considered undruggable. The most recent advancements in identifying and targeting mutations such as MET, exon 14 skipping, RET fusions, and ENTRGen gene fusions have further refined how we treat lung cancer. These discoveries underscore the importance of having reliable, accessible diagnostic tools to quickly identify these mutations. Real-time PCR plays a critical role in enabling this level of precision. Now, molecular diagnostics have fundamentally changed how we understand and treat lung cancer. Traditionally, histopathology was the gold standard for diagnosing cancer. While it's excellent for understanding tumor morphology, it tells us little about the genetic makeup driving tumor growth. The advent of molecular diagnostics, including techniques like immunohistochemistry or fluorescence in situ hybridization, began to address the gap by enabling the detection of protein expression and chromosomal rearrangements. However, these methods also have limitations in terms of sensitivity and scalability. Today, the technologies like real-time PCR and NGS allow us to dig deeper, identifying specific mutations in cancer genomes with high precision. Real-time PCR in particular stands out for its speed, affordability, and ability to scale, qualities that are essential for ensuring broader access to precision oncology. This shift has been critical in guiding targeted therapies and improving patient outcomes. In lung cancer, several genetic alterations drive tumor development and progression. Each of these mutations are only aids in the understanding of cancer biology, but also presents an opportunity for targeted therapies. Key mutations like EGFR, which is common in non-small cell lung cancer, and KRAS G12C, an important target for newly developed inhibitors. Fusion genes like ENTRG, ALK, RET, and ROS1 play critical roles in tumorigenesis and have specific inhibitors available. Lastly, MET exon 14 skipping mutations are gaining attention for their role in aggressive tumor behavior. Accurate detection of these mutations is essential for the timely and effective deployment of targeted therapies. To identify these mutations, we rely on a number of diagnostic tools. Immunohistochemistry and fluorescence in situ hybridizations are traditional techniques but have limitations in throughput and sensitivity. Next-generation sequencing, or NGS, offers comprehensive analysis of multiple mutations in one test. However, its high cost, long turnaround times, and infrastructure requirements can make it less accessible. This brings us to real-time PCR. While it may not detect all mutations at once like NGS, its speed, affordability, and accessibility make it a powerful tool for detecting specific actionable mutations. Today, we'll focus on why real-time PCR stands out. Real-time PCR, also known as quantitative PCR or qPCR, is a technique that allows us for amplification and detection of DNA or RNA in real time. And unlike traditional PCR, which only shows endpoint results, real-time PCR monitors the reaction as it progresses. This technology is not only highly sensitive and specific, but also remarkably efficient. From sample collection to final results, it can take as little as 2 to 4 hours. Its minimal sample requirement and ease of use make it especially attractive for clinical diagnostics, particularly in lung cancer. Real-time PCR offers several advantages, particularly for lung cancer diagnostics. Its speed means patients can receive results in the same day, which is critical for initiating timely treatments. Cost-wise, it is significantly more affordable than NGS, making it a feasible option for many healthcare settings. RT-PCR is also scalable, whether in a high-throughput reference lab or a small clinical setting. Most importantly, it is accessible even in low-resource environments where there is limited availability of instruments. While NGS is a groundbreaking technology, it comes with challenges. The cost alone can be prohibitive, especially in low and middle-income countries. Additionally, the time it takes to process and interpret results can delay critical treatment decisions. NGS also demands advanced infrastructure and specialized expertise, which not all labs possess. Furthermore, the vast amount of data it generates may include information that isn't clinically actionable, adding to its complexity. Now let's compare real-time PCR and next-generation sequencing in clinical context. While both are essential, they cater to different needs. Real-time PCR excels in speed and affordability, making it ideal for detecting specific mutations quickly. On the other hand, NGS provides a broader view, capturing all potential mutations in a single run. However, its high cost, long turnaround times, and infrastructure needs make it less accessible for routine diagnostics. Real-time PCR fills the gap by offering a practical solution for rapidly actionable, cost-effective testing. One of the biggest advantages of real-time PCR is its adaptability to resource-limited settings. Its affordability and relatively low infrastructure requirements make it ideal for use in regions where advanced technologies like NGS are out of reach. Real-time PCR assays can, for the most part, be run on any real-time PCR instrument as long as the instrument meets the technical specifications for the assay. For example, most EntroGen assays multiplex between 2 to 4 targets, so they contain probes labeled with common fluors detected by most modern real-time PCR instruments. So as long as the instrument can detect these fluors, the assays should work on them. For example, for EntroGen products, the majority of assays are already validated on most common instruments from Thermo Fisher, Bio-Rad, Roche, QIAGEN, but routinely support we as EntroGen, we routinely support users who run these assays on non-common instrument brands. And I've listed some of these here, Analytical Jena, Tianlong, and so on and so forth. Now, real-time PCR workflow is quite simple. It starts with nucleic acid isolation, followed by PCR plate setup, the PCR run on the instrument, and concludes with the with the data analysis. For nucleic acid isolation, we recommend use of commercially available DNA or RNA isolation kits. These can be either column or magnetic bead-based kits, either automated or manual procedures. The typical DNA input is 1 to 20 nanograms per reaction for most EntroGen assays, and most of these assays require multiple reactions per sample in order to detect a large number of mutations, as I will show you later on in the presentation. For RNA, it's a bit more, close to 40 to 80 nanograms. For quantification, we prefer using fluorimetric quantification because it is more accurate. This is things like qubit, for example, but spectrophotometric quantification such as nanodrop is also suitable. For quality assessment for DNA, all EntroGen assays contain an internal loading control that tells the user whether there is sufficient amount of DNA in the reaction. We also offer qPCR-based quantification assays that can be used to more accurately assess the amplifiable copies of each sample. Now, for isolation, usually it takes about 2 to 4 hours. It depends on the assay that's being used. Now, the PCR plate setup is very simple. Each kit is provided with the primer probe mix, reaction mix, and a positive control mix. The reaction and primer probe mixes are blended to make a master mix. Then these are then aliquoted into a PCR plate. Samples are added, plate is sealed, and it is ready to be run. This process takes approximately about 30 minutes. The runs are performed on a variety of real-time PCR instruments. EntroGen validates its assays on a number of these commonly found platforms, mainly instruments from Applied Biosystems, Bio-Rad, Roche, and QIAGEN. And runs here takes approximately 90 minutes to 120 minutes. Once the run is completed, the data analysis can be done using a PCR instrument software. Most EntroGen assays are quite easy to analyze because you're looking at the presence of a mutation based on a predefined CT value range. If the CT value meets a specific threshold, then the sample is positive for the mutation detected by the reaction. As mentioned before, we also offer software that can be used for data analysis. This software allows for automated mutation status determination based on the predefined criteria. The user imports the instrument data file after the run is completed, and the software produces a report. It shows whether the run is valid. I'm sorry, it shows that whether, I think I have an issue here with my slide, but it shows whether the run is valid, whether the samples are valid, and it also shows the mutation status of each sample. And the report looks like this. It contains the run information, such as the date, the run name, operator name, etc. The run control data, which shows if the run is valid or not, the sample mutation status, and the CT value for traceability. And the report, this report is locked and secure to prevent intentional or unintentional tampering of the data. However, it is an XML file format, which means that the data can be easily parsed and integrated with an existing laboratory information system. So just to recap what I've presented so far, speed and cost are critical factors in molecular diagnostics, especially for lung cancer. Real-time PCR offers a significant advantage here. With its ability to deliver results in hours, it enables clinicians to rapidly identify common actionable mutations like EGFR or KRAS, allowing treatment to begin almost immediately. But this doesn't mean that NGS has no role. It absolutely does. NGS provides comprehensive profiling that is indispensable for identifying rare or complex mutations. However, due to its long turnaround time and a higher cost, it is not always practical as a first-line test. For these reasons, the most efficient approach combines the strengths of both technologies. You start with a real-time PCR to quickly detect common mutations and reflect to NGS if no actionable targets are found. This workflow ensures that patients benefit from the speed of RT-PCR while leveraging the depth of NGS when needed. This complementary approach provides the best of both worlds, maximizing diagnostic efficiency and treatment outcomes. So looking at this proposed workflow, a sample is first tested with real-time PCR. If actionable target is identified, then an appropriate targeted therapy can be initiated right away. If a target is not identified, then NGS testing should be performed to find alternate targets. If found, the patient can be treated with an appropriate targeted therapy. Otherwise, they will be treated with non-target therapies. So shifting gears to some specific tests offered by EntroGen, we offer several real-time PCR assays that identify clinically actionable targets in lung cancer. These include EGFR kits, one for detection of mutations in exons 18 to 21 from genomic DNA from FFPE tissue, and the other for detecting the same mutations plus C797S resistance mutation from cell-free DNA from plasma, essentially for liquid biopsy version of the kit. As you may already know, there are different subtypes of EGFR mutations. The most common are the I think somebody is not muted. Okay. So the most common mutations are the exon 19 deletions and L858R mutations, which account for approximately 85% of the mutations in EGFR and lung cancer. There are several approved drugs that are used to treat lung cancer patients whose tumors harbor these mutations. Less common are the G719X mutations, L861Q, and S768I mutations. These account for up to 10% of the EGFR mutations in lung cancer. There are some second-line EGFR TKI therapies targeting these mutations as well. And lastly, there's a lot of focus on exon 20 insertions in EGFR, which account for up to 7% of EGFR mutations. So the EntroGen mutation analysis kit is for detection of mutations in exons 18 to 21 of EGFR gene, including the T790M resistance mutation, which confers resistance to first and second-line EGFR TKI therapies. This kit detects all of the mutations that I had mentioned in the previous slide. This assay requires genomic DNA isolated from FFPE. However, I do want to mention here that although the assay was validated on genomic DNA from FFPE tumors, there was a publication in 2015 by a user where a clinical laboratory demonstrated successful usage of this assay on DNA isolated from cytology smears. So this is quite a robust test that can utilize DNA from various sources. The assay requires running 8 reactions per sample with approximately 5 nanograms of DNA required per reaction for a total of 40 nanograms. Detection is done with 2 floors, FAM for the target variant and VIG for internal loading control to ensure there is sufficient amount of DNA in the reaction. This assay is used for detection of EGFR mutations in DNA isolated from FFPE tissue. Now, given that there are 8 reactions per sample needed to be run on this assay, it fits quite nicely on a column of a 96-well plate. So each column represents a sample. So taking one lane for positive control another lane for negative control that leaves up to 10 lanes for samples. So up to 10 samples can be tested at once on each run. The total time for once you have DNA for sample to be tested from sample to result is approximately 2 hours. We have another EGFR assay specifically designed for liquid biopsies. It uses cell-free DNA isolated from plasma. This test has also been validated for use with genomic DNA isolated from FFPE tissue as well. The assay detects an additional resistance mutation in reaction to the C797S mutation that is not part of the previous tissue kit. This mutation is important due to the resistance to third-line EGFR TKIs. It's been shown that approximately 40 to 50% of patients treated with third-line EGFR TKIs eventually relapse due to the tumors acquiring this resistance mutation, which prevents the drug from covalently binding the target protein. Typically, the C797S mutation is not tested in patients who have not yet received EGFR TKI therapy because it is not present in untreated lung tumors. So this test would be most suitable for testing tumor samples after EGFR TKI failure. The first reaction of this assay is highly multiplexed to detect the T790M, L858R, and exon 19 mutations. In total, there are 44 mutations detected in this reaction, in this one reaction. This assay was developed in this manner to minimize the input requirements, given that this was designed for liquid biopsy, where cell-free DNA quantities can be quite low. This assay requires approximately 1 to 5 nanograms of cell-free DNA from plasma. The detection for reaction 1 requires 4 filters, FAM, ROC, Sci5 for the mutant targets, and VIC for the internal loading control. The other reactions require just 2 detectors, just FAM and VIC. Since there are 7 reactions for this test per sample, again, similar to the tissue kit, this also takes up almost a full column, so just 7 instead of 8. So taking 1 lane for positive control and another lane for negative control, it leaves up to 10 lanes for samples. The total time once you have DNA from sample to the result is also about approximately 2 hours. So just to recap the liquid biopsy workflow, plasma is spun out of whole blood. And we do recommend using Streck tubes or Paxgene tubes to stabilize the white blood cells in order to prevent contamination of the circulating tumor DNA with genomic DNA from lysed white blood cells. Then the cell-free DNA is isolated from the plasma using a commercial isolation kit. EntroGen does offer a cell-free isolation kit as well for this purpose, followed by mutation analysis using the provided reagents with the PCR test kit. Now, having discussed the importance of EGFR mutations and their role in lung cancer, let's shift our focus to another significant driver mutation in lung cancer, which is KRAS. While EGFR mutations are prevalent, KRAS mutations actually occur even at a higher frequency in lung cancer. In fact, they are found in approximately 40% of lung adenocarcinomas, making KRAS a critical target for both diagnostic and therapeutic strategies. Understanding KRAS mutations is essential as they present a substantial portion of the patient population and have implications for treatment options and outcomes. Here I'm showing the distribution of different KRAS mutations within the KRAS mutated population in lung cancer. The G12C mutation is the most common, representing approximately 46% of cases. Historically, KRAS mutations are considered difficult to target, often described as undruggable. However, in recent years, significant progress has been made in developing targeted therapies, particularly for G12C mutation. Currently, there are 2 drugs that have been approved that target G12C used for treatment of lung cancer, when the mutation is present, that there's a number of active clinical trials for various pharmaceutical companies developing more drugs that target G12C and other G12C mutations. So testing for KRAS mutations in lung cancer is crucial, particularly for identifying the G12C variant. This ensures that patients who can benefit from targeted therapies receive the most appropriate treatment. Now, EntroGen offers a comprehensive KRAS kit, and which can be used to identify KRAS mutations in genomic DNA isolated from FFPE lung tumor specimens. This assay requires 12 reactions per sample if all KRAS mutations in exons 2, 3, and 4 are desired to be tested for. But one thing to keep in mind here is that this assay was originally developed for colorectal cancer, where the distribution of KRAS mutations is different than those in lung. In lung cancer, as I had shown in the previous slide, a significant portion of KRAS mutations in exon 2, specifically codon 12 and 13. So one shown in the previous slide. You can imagine that one could potentially get away with testing only for the first 7 reactions that cover codon 12 and 13 mutations in order to minimize the input requirements for the assay and potentially increase the assay's throughput. Again, this assay multiplexes the amplification of the mutation target and internal loading control detected with FAM and VIC filter sets, respectively. Looking at the plate layout for this KRAS test, if all 12 reactions are run for the sample, then each sample takes up a whole row, allowing for only 6 samples to be tested per run. However, if just the first 7 reactions are run for this assay for detection of only codon 12 and 13 mutations, which are primarily the KRAS mutations found in lung cancer, the plate layout would look like this, and up to 10 samples can be tested on each run. Now, however, please note that the data I showed on the previous slide reflects KRAS mutation prevalences in Caucasian population only. It is important to note that mutation distributions can vary significantly across populations. When implementing KRAS testing strategies in Indian population, one must consider their specific mutation distribution to ensure that testing approach is tailored appropriately to accurately diagnose for an effective treatment decision. Now let's focus on BRAF mutations in lung cancer. These mutations are found in roughly 1 to 5% of lung adenocarcinomas. Among these, the most common variant is BRAF V600E, which constitutes about 50 to 60% of all BRAF mutations in this context. Non-BRAF V600E mutations, such as the G469A or D594G, are also observed, but they carry distinct clinical implications compared to V600E. For BRAF V600E in particular, is particular to note because it has been shown to respond well to targeted therapies such as BRAF and MEK inhibitors. So identifying BRAF mutations in lung cancer is therefore a critical approach, and it helps guide therapeutic decisions. EntroGen's BRAF test has been validated for both DNA from FFPE as well as cell-free DNA from plasma. It requires running 5 reactions per sample to detect V600E, E-complex, K, D, R, M, and G mutations, all in the codon 600. This assay was originally developed for use in melanoma samples where the non-V600E mutations are present. However, in lung cancer, they have not been shown to be present. When running all 5 reactions for this test, we recommend loading them in this manner, so up to 14 samples can be tested at once. And again, the total runtime for this assay is about 2 hours. Now let's focus on some fusion genes in lung cancer. So fusion genes, MET exon 14 skipping mutations represent a significant area of focus in molecular profiling for lung cancer, particularly non-small cell lung cancer. Although these genetic alterations represent a small percentage of lung cancer population, ALK rearrangements have the highest prevalence, ranging from 3 to 7%, while the others occur less than 5% of cases. Despite their rarity, they present significant lung cancer subtypes that can be effectively treated with specific targeted inhibitors. In recent years, several new drugs have been approved to specifically target these fusion genes, and these are shown here on the slide here. Now, EntroGen offers 2 assays for fusion gene detection in real-time PCR. The first is a lung cancer RNA panel, which is for detection of fusions in ALK, RET, ROS1, as well as MET exon 14 skipping. And the other assay is for detection of NTERC mutations. Because the input for these assays is RNA, cDNA synthesis is required prior to amplification of the targeted sequences. Both of these assays utilize a one-step reaction master mix that performs the reverse transcription step and PCR amplification in the same reaction. So this significantly improves the workflow for RNA-based assays and reduces not only the hands-on time, but also risk of contamination and sample mix-up. The lung cancer RNA panel requires 8 reactions to be run per sample. The input is RNA of approximately 80 nanograms per reaction. It is a multiplexed assay with FAM and Cy5 detectors used for fusion gene variants, and VIC for internal loading control. This assay detects 47 fusion variants, and its analytical sensitivity is as low as 10 copies per reaction. Again, with 8 reactions per sample, up to 10 samples can be tested on one run. Even though this assay incorporates cDNA synthesis, the reverse transcription step, it still takes approximately about 2 hours to run. The enteric fusion gene detection kit also requires RNA as input into 8 reactions per sample with approximately 80 nanograms per reaction. It uses 2-color detection, FAM for fusion genes and VIC for detection of internal loading control. So it detects these mutations in this manner. As you can see, reactions 1 through 3 detect the enteric 1 fusions, 4 through 6 enteric 2 fusions, and 7 and 8 enteric 3 fusions. The assay does not distinguish between fusions detected within each reaction. And the assay detects 65 enteric fusion variants with an analytical sensitivity of as low as 25 copies. Now, similar to RNA panel layout with 8 reactions, one can run up to 10 patient samples on a single run. Now, I want to conclude by showing this workflow again from an earlier slide. And I want to emphasize the important point here. We are not suggesting that real-time PCR is a complete replacement for NGS. Instead, these technologies complement each other. Real-time PCR testing provides rapid results at a fraction of the cost and labor compared to NGS. By using real-time PCR as the first step, actionable targets can be quickly identified and treatments initiated without delay. For cases where real-time PCR does not detect an actionable target, NGS serves as a powerful follow-up tool, offering a comprehensive analysis to uncover alternate targets. This approach ensures patients receive the benefit of both technologies, speed and cost effectiveness from PCR, and the depth of analysis from NGS. Thank you very much for your attention. I can now take questions if there are any.
Thank you, Dr. Gary, for your time. And now I would like to request the customers or whoever is attending the webinar, in case you have questions, please do unmute yourself and ask them. Or alternatively, you can also put your questions in the chat box. As I can see, Dr. Gary, there is already one question which has been posted by Dr. Imran. He's asking what can be interpreted if there is borderline CT or delta CT value or inclusive results come by RT-PCR technique, which is most common sensitivity issue in this technique. Also concern of DNA quantity issue from FFPE sample in this multi-tube RT-PCR panel. How to overcome by using this technique instead of NGS?
Okay, so of course, yes. So sometimes with real-time PCR, you do get borderline signals. And the way we typically suggest to overcome this is to increase the input. So with PCR, if let's just say that the borderline sample has, or not a borderline sample, but a sample gives a borderline CT value. If this CT value is due to non-specificity, which in some cases PCR assays do produce non-specific signals. If it is due to non-specific amplification, then increasing the load, the input of the sample, should not affect and this is what we normally see in our validation, in our design and validation of these assays. So if you increase it, the CT value for the target signal should proportionally increase. So it should, if the loading control, let's say for VIC, increases by half a CT, the target CT value for FAM, for example, should also decrease by half a CT. And that typically indicates a true signal, a true mutation signal. If it does not change, and in some cases increasing the input will actually eliminate this nonspecific, if it is a nonspecific, because of competition with the internal control reaction, then it goes away and it tells you that this is a nonspecific signal. And the second question I believe it was regarding fragmentation of DNA in FFPE tissue. Is that
Yeah,
correct?
the question was also concern of DNA quantity issue from FFPE sample in this multi-tube RT-PCR and how to overcome by using this technique instead of NGS.
So with RT-PCR, we typically design our assays to be able to identify, to be able to amplify from very short, very small fragments. So we take, of course, we understanding that these assays are being used on FFPE, which is typically very fragmented. We do design them to be less than 120 base pairs in size. And that typically gives us a fairly good chance of being able to amplify from heavily fragmented samples. So this is what we've typically found with these PCR assays. If it looks like the internal control gene tells you that the sample is underloaded, again, we suggest to increase the input to be able to bring it in range. And in most cases, our users are able to bring it in range to be able to use it, to be able to accurately call the mutation.
Thank you, Dr. Gary, for your answers. Anyone else who has any question that they would like to ask? I don't see any further questions or hands being raised. I think there is one from Dr. Anil. Dr. Anil is asking, I was one of the initial users of EntroGen assays for KRAS, NRAS, and QRT-PCR for BCR-ABL fusion transcripts. Real-time PCR is not ideal for lung cancer evaluation, where the biopsies are usually needle cores and DNA quantity extracted even from well-fixed FFP tissue is an issue.
So again, it really depends on clearly the protocols used for the tumor biopsies. You know, from our experience, the usage of our EGFR, for example, or any of our lung cancer assays, we know that some of these assays are actually our highest used assays globally. We typically don't hear a lot about insufficient amount of DNA that they get to use with these assays. You know, it's a little difficult to answer this question because it really depends on the institution and how they are isolating DNA, how they're collecting the sample, and how they're isolating DNA. We've shown that, and others have shown that even from cytology smears from lung cancer samples, they're able to they're able to isolate enough DNA to be able to identify mutations. And there was a publication that I had posted in one of the slides there that showed that they are, in fact, able to identify mutations in such small amount of sample. So it's difficult to answer this question without knowing a little bit more information about how much DNA they're actually getting. So I'm not sure if the user was loading the amount of DNA that's specified in the user manual for the kit, for the EGFR kit, for example, or have they tried loading less amount, because there is a range of DNA input that we specify that's needed. But the internal loading control typically will guide whether there's sufficient amount of DNA in the sample.
Dr. Gary, there is one more question from Dr. Imran. He's asking, what was the sensitivity of the cfDNA by using RT-PCR technique, and how is it different from ddPCR? Digital droplet, I believe.
Right. So, you know, for the ctDNA assay that we have for with real-time PCR, we were actually able to get down to, and I believe this is in the user manual as well, disclosed from our validation data. We're able to get down to as few as 10 copies. We don't present our sensitivity in percentages because we believe that presenting the analytical sensitivity of these liquid biopsy assays in terms of percentage is misleading. And here's the reason. If I see this quite a lot in brochures from especially digital PCR, droplet digital PCR assays, where they are able to detect 0.005 or 0.001% mutant over wild type. When you really back calculate the number of copies that's required to be able to get down to that level of sensitivity, you really need to have, you'd be loading hundreds of nanograms of cell-free DNA, which we all know is not really feasible. You would have to bleed the patient to be able to get that much DNA, cell-free DNA from patients. So when we did our validation for our cfDNA assay for real-time PCR, we quantified the number of copies and tried to detect the fewest number of copies that the assay can detect. And we were able to get down to, I believe, approximately 10 copies per sample. And that's 10 copies per 2 ml plasma sample. So I don't know how directly that compares with ddPCR because we did not do that direct comparison. My experience with ddPCR is solely based on the marketing documentation and the brochures that manufacturers of the PCR assays provide.
So, Dr. Gary, there is another question from Satyendra. He's asking, is it recommended to use the EPAS software for analysis on the ABI platform?
Yeah, so EPAS, which stands for EntroGen PCR Analysis Software, actually does work with ABI instruments. We have a number of these. So ABI 7500, QuantStudio 3, QuantStudio 5, and the various QuantStudio instruments, of course. They are all compatible, so they all produce a compatible EDS file that is then imported into EPAS software, and it does the analysis, basically automates the analysis. I do recommend using EPAS software. It does streamline the process for data analysis. If you don't find your instrument in the list so when you install, again, EPAS is provided free of charge to our users of our kits. So if you installed EPAS and you do not see the assay that you're looking for, the instrument that you're looking for the assay, for the data that you're trying to analyze, then just simply ask us, and we will provide a patch file that will include the assay and the software. Now, there are some limitations with EPAS. EPAS is currently only designed to analyze only 2-color assays, so FAM and VIC. So assays such as lung cancer panel, lung RNA panel that has more than 2 colors, that will not be currently available. We're working on that, but it's not currently available to be analyzed. So there are some limitations, but for the most part, yes, absolutely. With ABI instruments, you can definitely use EPAS, especially for EGFR or KRAS or BRAF.
Thank you, Dr. Gary, for such an I think there is one more question from Dr. Imran. He's asking, I am more interested in cfDNA by RT-PCR. Do you have any positivity during your validation? This is just for my knowledge.
Can you clarify what you mean by positivity?
Dr. Imran, would you be able to unmute yourself and ask this question directly? Hello?
Is the question whether we found positive samples in our data set during validation or something else? I just want clarification on the question.
I'm not sure if he's able to unmute himself. So what we can do is I can ask Dr. Heather connect with him later on and maybe get more details on his questions so that he can interact with you about this.
Okay, no problem. Yeah, I'm more than happy. This assay was actually there was actually also some poster presentations, and I believe there's some publications as well using this assay. It is a widely used diagnostic test here in the US and elsewhere, as well as in Europe. And I believe there are some publications, peer-reviewed publications also on this assay. But I'm more than happy to provide additional information if you can send me an email with it with the
Sure,
questions.
we'll connect with him later on. So
Yes.
I would just like to thank you, Dr. Gary, for this insightful and engaging session. Your expertise and this deep understanding of this subject have provided valuable insights into the advancements of the work that EntroGen is doing. And we sincerely appreciate you taking the time, especially considering the hour on your side of the world, to connect with us here in India. Also, a heartfelt thanks to all of our attendees for your active participation and thoughtful questions. Your enthusiasm and engagement has truly made this session so rewarding. Finally, I would like to express my gratitude towards EntroGen for their continued partnership and innovation in molecular diagnostics, and to the marketing team of DSS for organizing this webinar. For any further questions or follow-ups, please feel free to reach out. You may send me an email or to the marketing team or whoever is your connect in DSS, and we will reach out with the answers required. Please stay tuned for any further updates coming up from our team for further webinars. Thank you, everyone. Have a great day ahead. And to you, Dr. Gary, wishing you a
Thank
very
you
good
very much.
evening.
Thank you very much for the opportunity. I really appreciate that.
Thanks a lot. Thank you.
Thank
Bye-bye.
you. Bye-bye.
Bye, everyone. I'm
Dr.
ending the
Gary.
session.
Bye-bye, everyone.